Hello, everyone. Greetings to everyone from all over the world who have joined for the symposium today. I'm Priyanka Baloni, a research scientist in Hood Price Group at the Institute for Systems Biology. It's my pleasure to have all of you here for the symposium. And I'm delighted that so many researchers from all over the world have Zoomed in yesterday and also today to participate in our course and the symposium that is being hosted by ISB. I'm so excited for the amazing lineup of speakers that we have today for the symposium. And as Sean mentioned, we have 3 different sessions. This is the first session in which the theme of the session is mechanistic models. And we have Professors Jason Papin and Ines Thiele, who will share their research done in the group on probing metabolism in microbiome. So while I'm introducing Professor Jason, I would request Jason to start sharing his screen and we can then start the session. Professor Jason Papin is a professor in the Department of Biomedical Engineering at the University of Virginia. After his training in bioengineering at the University of California, San Diego, Jason joined as a faculty at the University of Virginia in 2005. His lab works on the problems on systems biology, metabolic network analysis, infectious diseases, toxicology, and cancer. His group has also developed numerous computational approaches for integrating high throughput data into computational models. Jason is the co-editor-in-chief for PLOS Computational Biology and an editorial member of Molecular Pharmacology and Cell Systems. It's always a pleasure to listen to Jason's talk. Today, Jason will talk to us about metabolic mechanisms of interactions in microbial communities. Without further ado, please join me in welcoming Professor Jason Papin.
Thank you, Priyanka. So you can see the right screen and everything, right?
Yes.
Okay, thanks. Yeah, so it's great to be here, great to be able to participate in this symposium, and great to do so from my own home. That's kind of awesome. I'm on the faculty at University of Virginia, Department of Biomedical Engineering. Excited to be a part of such a great program today. I just want to first start off, just give a one-slide introduction of my group, different kinds of research activities. We have 5 primary research activities. One, we do a lot to develop computational methods for integrating high throughput data to create these predictive metabolic models for a variety of different applications. But one facet of the group is the focus on development of methods and algorithms for taking that data to create these predictive models. One of the primary application areas we focus on is trying to understand metabolism of microbial pathogens. And we use these models to identify drug targets, understand how metabolism is supporting the evolution of antibiotic resistance, for example, understand the evolution of metabolism in a microbe during the course of a chronic infection. And every once in a while, there are opportunities for developing simple, high throughput types of experimental technologies focused on phenotypes and readouts that are related to metabolism. This happens on occasion. In full disclosure, some of this has been licensed to a biotech company that I'm a part of. Then a fourth category of activity in the lab is extending some of these metabolic network models to look at a variety of different biomedical challenges and focusing on network models for rat, human, various mammalian systems. But what I'm going to focus on today is how we use these tools to try to characterize the metabolism of communities of microbes. And there are 3 brief little stories I'm going to share during the time that I have today. One, I'm going to briefly introduce what we like to call a model microbial community. Model systems are critical in biology, and there aren't really a lot of good model systems when we think of microbial communities. So I'm going to introduce one. I'm going to talk about some work that we've done to delineate the metabolites that are exchanged between members of a microbial community. We know metabolism is important as a part of these microbial communities, but it is an inherently very difficult problem because we're talking about multiple species interacting with each other with a shared pool of metabolites that they can be contributing to or drawing from. So how do we characterize the metabolites that are important in the exchange, in the interactions between microbes? And then the third story I'm going to talk about today is some new algorithm method development for how we map measured metabolites in a complex community to specific species in that complex community. So that's where we're going to go today. Now, first, let me talk a little bit about a model microbiota that's called the altered Shadler Flora. Now, the idea of this community, altered Shadler Flora, has been around for several decades. And the history of this is back in the 60s, 70s, there was some work where some microbiologist, Shadler, took feces from mice and would culture the microbes that were present. And after lots of work culturing and identifying overly abundant microbes in these samples, he defined this community of microbes that's now referred to as the Shadler Flora. Now, the idea here is that when you grow a mouse without any bugs, so it's in a germ-free environment, it develops an abnormal immune system, among other abnormalities. But with the Shadler Flora, this community of 8 microbes, introduced into a germ-free mouse, so this is a gnotobiotic mouse, that mouse develops normal-ish immune system. It's still not quite exactly normal, but the connection between a defined community and its ability to restore a fairly normal immune system in a mouse was really what motivated the development of this community. Now, we haven't talked much about this community. Jackson Labs, other companies that provide mice, they will often use the Shadler Flora to try to standardize the microbiota of mice in the colonies that they have. But over the last couple of years, we've done a little bit of work to try to better characterize this community. And this community, you see images there of these 8 microbes, first developed in the 60s by Shadler. It was refined and adapted from that original cocktail in the 70s. That's why it has the name altered. Importantly, all 8 of these bugs are culturable, and the impact on mouse physiology has been evaluated. Now, back in 2015, 2016, there's a group, Michael and Gregory that you see there from Iowa State, who sequenced the genomes for the first time of these 8 bugs. But that was kind of it in terms of real good molecular characterization. So with a gift from Michael and Greg, we began to do a little bit of analysis of this community. Now, first, this work was published in ISME in 2017. And first author is Matthew Biggs. So you see his picture there. He now works with a biotech company in North Carolina. But just to kind of summarize a paper of some of the characterization that we had done of this community, I want to draw your attention to this data. What you see here on the, what was done was we took some previously published data where there were 15 different mice, wild mice, and the communities of microbes in the gut of these mice was characterized and sequenced. And we looked at the overlap between the functional categories of genes that the ASF, those 8 species, comprised, with the functional categories of genes that were present in subsets of these microbial communities from these 15 wild-type mice. And I just want to draw your attention to the last point here. So this is across all 15 of those microbial communities, okay? What fraction of these NOGs, and these are clusters of sequences where the proteins in that cluster share the same function. So what fraction of the NOGs represented in all 15 of those microbial communities were captured by the ASF community, as well as random draws of 8, 16, and 32 species from those metagenomes. And data is a little messy here, but just to focus on the arrows here, this is the median, and then the range over which the fraction of NOGs that were captured in all 15 for 8 random draws, 16 random draws, and 32 random draws from that community. An important point I want to emphasize here is that the ASF, in terms of the functional categories that the ASF captured, was equivalent to any random draw of 32 species from that microbial community. So all of this just to emphasize the point that the ASF community serves as a fairly good surrogate for wild type microbiota functionality. Now, this was the beginning then and the first characterization of this model microbial community. But there's this important question of, we know that these genomes may hold good content in terms of functionality, but how do we characterize how each of these bugs are interacting with each other? Now, a paper published in my group a few years ago, and just in disclosure, some of this technology has also been licensed out. But this was a paper where Tom Motino, a PhD student in my lab, led the development of a Coculture plate. So this plate has the same outer dimension as a normal 96-well plate. But as you see, there are pairs of wells that have this membrane that's vertical. So think of a transwell plate, except where that filter is rotated, that allows 2 different kinds of communities to interact with each other. And I'm not going to elaborate more on this, except just to say that in the paper, we did a lot of the proof of concept to show that you could grow separate communities of microbes, allow the exchange of metabolites, and do it in a way, because the vertical is transparent, where you can measure optical density and as a surrogate of growth as these communities are exchanging metabolites with each other, and the ability to separate out physical dependent and independent interactions, okay? So that's an example of some kind of technology, but still there's this question where you have data maybe, or you have microbes that are growing as part of some kind of shared community, but we want to understand what's the mechanistic relationship in how metabolites go from one to another. So in order to address this question, Greg Medlock, a PhD student in the lab, who's now a faculty member in pediatrics at University of Virginia, he developed this strategy. And I'm going to walk through the steps here. First thing, he collected a set of data. So OD measurements, strain-specific qPCR, as well as metabolomic profiling under 3 different conditions, where we grow a bug in a culture by itself, another bug in a culture by itself, and another culture where they're grown together, okay? Measure growth, as well as the metabolome, the secreted metabolome of each of these 3 conditions. Now, first set of data I want to talk about is the synergistic or antagonistic growth that can happen when the bugs are grown in coculture. And what you see here in this data, I mean, a little dense. So on this axis, we have strain 1 abundance, the Z-score, okay? On this axis, when the cultures are grown by themselves for strain 1, and for strain 2, when it's grown by itself. And then what you see in these open circles is the relative abundance of strain 1 and the relative abundance of strain 2 when they're grown together, okay? Now, and that's what the interpretation is here. Now, to interpret some of the coculture results, when you see the cocultures exist in this quadrant, what it means is that the strain 1 is more abundant than when it grew by itself, and strain 2 is also more abundant than when it grew by itself. So this is a mutual relationship. When you see in the coculture, you see data fall into one of these other 2 quadrants. There's a parasitic relationship where one has a growth benefit and the other is, it loses its growth capability. And this is where there's a competitive relationship. Similarly, if you're along these lines, there are mensal, commensal, and other types of relationships. So for the altered Shadler Flora community, this is the breakdown of the kinds of relationships that we see. We see a distribution of mensal, competitive, parasitic, and commensal relationships, but there are no pairs for which there's a mutual relationship or neutral, okay? And this just represents kind of the scale of whether or not a given microbe is less or more abundant. What the white dots mean is that there's a p-value less than 0.1 for that particular growth outcome. Okay, now this is the first set of data characterizing the kinds of growth dependencies and relationships between the members of this community. But what we want to get at is how is the mechanism, what are the metabolites that are enabling or facilitating or a part of those growth relationships? So the next set of data we needed was the metabolomic profiling of the mono and co-culture supernames. And what you see there is a heat map of a collection of metabolites that we measured in the monocultures, and whether or not that metabolite was consumed in blue or produced in red versus the baseline and the starting media. So we processed this data, and just to show you how this data looked, you see here for the monocultures and co-cultures of two of the species of this ASF community, you see again, this is the principal component, the first component on the x-axis, second on the y. Now, the co-culture metabolome is very distinct from the two monoculture metabolomes. The monoculture metabolomes are very independent of each other. Co-culture is right in the middle. And it's the separation that's the co-culture versus monoculture. But still, the question is, what metabolites are driving these growth interactions? So we have growth data, we have the metabolomics data, and now to get at that question, what are the metabolites driving those interactions? We developed this approach here, which we call CONYE, which stands for a constant yield expectation. Let me just step through the math a little bit here. It's a very simple test. So we have C sub EI. This is the expected abundance of a particular metabolite in co-culture is going to be equal to the sum of the abundance of the strain, of one strain in that co-culture with the yield of the metabolite in that culture, plus the expected abundance of the other strain in the co-culture times the yield of that metabolite in the monoculture, plus the abundance of the metabolite in fresh medium. So simply, we expect the amount of metabolite in the co-culture to be the sum of how much it's in the monoculture of one normalized to the amount of that bacterium, how much of that metabolite's in the other monoculture normalized by the abundance of that strain in that co-culture, plus what's in the media in the first place, okay? Now, so if we have a certain abundance expected from metabolite in one monoculture, in the other, the test is the observed co-culture abundance of that metabolite different than the predicted abundance of that metabolite in the co-culture? And that simple statistical test can help inform whether or not that metabolite is greater or less than you would expect in that co-culture presence. Now, let me give you an example of some of the data here on that point. We have here 4 different metabolites, lactose, lactate, valine, and proline. And in each square, what this is indicating in the upper right triangle, this is the expected abundance of that metabolite, and this is the observed abundance in that metabolite. And so for lactate, for example, there are several different pairs of ASF species, so of that Shadler Flora microbe, where the amount of lactose is less than expected. So it's consuming that metabolite, lactose, more than you would anticipate from the data in the monoculture. For some metabolites like valine, you see transitions between sometimes the metabolite is produced more than expected, sometimes it's consumed more than is expected. Now this, let me just summarize the data, kind of this overview, and then go to a specific example of what we're talking about here. of 1,290 metabolite co-culture groups for which we could predict some level of the metabolite abundance, the null hypothesis was rejected in 500 of those. So about 40% of the metabolite co-culture data is different than you would expect, suggesting that co-culture truly alters metabolism of a significant fraction of these metabolites. So let me dive in a little bit more specifically here. We have here, of the metabolites, the green dots represent different metabolites. We have across all the ASF pairs, these are metabolites where they're more abundant than expected. And on this axis is the statistical significance, so a decreasing p-value. So we have some metabolites that are more abundant than expected, others that are less abundant than expected. And this is of the subset of metabolites for which in the monocultures, they were produced by one species and consumed by another. So what's the interpretation of that? Well, for metabolites that fall into this category, there's a couple different hypotheses here. Perhaps the one species is consuming it more, or perhaps the other species is producing it less. For metabolites in this category, perhaps the one is consuming less and the other is producing more. And you can similarly generate these different kinds of mechanistic hypotheses for the collection of data that you have about how the abundance of the metabolites are different in the co-culture than you would expect as a sum of the monocultures. So I'm gonna give you a very specific example. Two different microbes here in that Shadler Flora have a numbering system, which you can look up if you like, but there was a particular pair for which there was evidence that alanine and proline were being produced by one species and consumed by another. Now, and that the consumer of those metabolites had improved growth than would be expected, okay? Now, what's interesting about this is a classic pathway associated with stickland fermentation in Clostridial species. So the survey of, again, expected versus observed metabolite levels suggested that 519 was a producer of metabolites that are frequent substrates for stickland fermentation, for which this other microbe could consume for a growth benefit, okay? Now, that's what that survey of the different metabolite levels, running it through that Kanye test allowed for. So we wanted to test that. And the experiment we did, we have here, this is the relative density relative to supplement, so how much growth this particular microbe is exhibiting under a few different media conditions. So this is just in the baseline media. And this is when we provide proline, alanine, isoleucine, and valine alone. And there's no statistically significant difference in the growth benefit. However, when we provide the proline and alanine together, there's a significant growth benefit for, again, 356, when those two metabolites are provided together, where there is no difference when other amino acids are provided in other pairs. So this suggested to us that this stickland fermentation was indeed facilitating growth of this ASF species 356, okay? It wasn't just the availability of the individual metabolites, but providing those metabolites as a pair. Okay, that's one story. This was published. I'll point you to that paper. What I want to do with the remaining time that I have is talk a little bit about this next challenge. So to do this, let me first just give a little background on some metabolic network modeling that my lab and others here attending the symposium are very engaged in. And just to give a brief introduction to some of the fundamental basics for metabolic network modeling at a genome scale. First step often involves mapping genes to associated proteins and reactions that those proteins or enzymes will catalyze. This allows you to delineate what are the functions that the genome captures from a metabolic functionality perspective. Second step involves constructing what's called a stoichiometric matrix. This is mapping the stoichiometric coefficients for the substrates and components of a network as rows, and then in columns, the different transformations that convert substrates to products. Once we have that matrix formalism in place, there are a variety of different matrix linear algebra tools, linear programming, other optimization strategies that allow you to generate a variety of different kinds of predictions. Now, for this kind of framework, we often assume that there's equal access to all the functionality in this network, okay? But we know that's rarely, if ever, the case in biology. So methods have emerged that allow us to integrate expression data to delineate the functions that are active in one state or one cell type versus another. Some of these methods have been developed by people here at this symposium. Now, these approaches are readily extended with high quality metatranscriptomic data, so transcriptomic profiling data in microbiomes, where we can, when that transcriptomic data is of sufficiently high quality, where we can assign from a large pool of transcripts which transcript level is associated with each species in a complex system, in a complex community, okay? But many of those methods involve delineating what are the exchanges or what's allowed to come in and out of that particular species or of that particular cell. But in microbial communities, we have a pool of shared metabolites. And so it can become difficult to constrain which are the substrates that one species is consuming and another's producing, given this pool of shared metabolites. So a lot of expression data integration algorithms require that you define those kinds of constraints, but this can be difficult to do. So in part, to help address this challenge, Matthew Jenner, you see his picture in the bottom right there, he's a postdoc in my lab. He led the development of this approach we call Riptide. Now, I'm gonna point you to the paper. This just came out recently for the details, but let me just give you kind of a sense for the method. So this method does not require that you constrain those exchange fluxes. Instead, what it does is two key components. So one, this method takes transcript data, and where this blue line represents the abundance of transcripts, where the red line represents a weight that you're going to put on the corresponding reaction. So simple idea, if the expression level is particularly high, then we're going to allow for flux through that particular reaction. If the transcript levels are really low, then we have a strong preference to not push flux through that reaction, okay? Now, other methods have done this. The important development with Riptide is that we also pair it with an assumption of parsimony of the network enzyme usage. And the evolutionary argument here is that the cell isn't going to be using energy to express things that it doesn't need to use, okay? So this integration of mapping transcript abundance with assumption of parsimony allows you to reduce some of the assumptions that typically go in these gene expression integration algorithms. Now, just to give you kind of a graphical illustration of this method, let's say, for example, we have a cell and there's two different pathways here, two different pathways of metabolism. Glycolysis is just a couple of steps, and that's one option where with the consumption of glucose, we can produce ATP. But an alternative pathway might be something like stickland fermentation, where I'm consuming two different substrates to generate or satisfy my ATP demand. Now, this pathway is less parsimonious, okay? There's more steps involved, more enzymes that would need to be synthesized. But if my transcript data indicates that these are highly expressed in comparison to the others, I can break that parsimony so that this becomes my prediction of a metabolic state, okay? Now, I'll just point you again to that paper on which Matt is a first author. There's several different analyses that were done to illustrate, offer some validation and confirmation of the validity of the approach. This is some example data where he integrated with riptides in an E. coli model some gene expression data associated with LB aerobic, some defined media, both aerobic and anaerobic, defined minimal media, and where we predict genes that are essential, non-essential, et cetera, and the categories of genes that show up as essential and non-essential are as you would anticipate given the different media conditions that they have. And there's additional validating information that I'll point you to the paper to help understand. Now, most recently, for some unpublished work, Matt has been extending this approach, again, back to a microbial community that has some clinical relevance. And his focus has been on understanding Clostridioides difficile. Now, this is a bug that is just a tremendous healthcare burden in the United States, 500,000 cases a year, approximately $5 billion healthcare costs associated with this. And infections arise when there's a disruption of a healthy gut flora. People are taking antibiotics, wipe out a lot of the healthy bugs that are in the gut. C. difficile will thrive in that environment. Now, there's a lot of interesting recent information that's coming out about the role that metabolism plays in the course and the process of infection with C. difficile. And I'll just point you to this recent preview that Matt wrote up that was posted in Cell Host Microbe just a couple weeks ago, where he summarized a couple of recent papers, which was the actual interesting discovery and work, demonstrating how C. difficile capitalizes on the host environment metabolically in order to facilitate the process of infection. So that preview, I think Matt did a good job summarizing some of that recent work. So Matt constructed a recent genome scale model of C. difficile. There have been a handful of others, but just to summarize, he was able to curate around, and I just included a list of a variety of different metabolic pathways that he curated in his recent reconstruction. He also ran through a variety of different validating data on gene essentiality, as well as some growth data that Matt has been able to compare model predictions with, just again to demonstrate the quality of the C. difficile model that he had built. Now, let me integrate this brief introduction, this new C. difficile model with the riptide approach for expression data integration in a clinically relevant setting. So in collaboration with a colleague, Rita Tamayo, who's at University of North Carolina in Chapel Hill, she recently discovered this interesting phenotype with C. difficile, where under certain growth conditions, and she did this, there's this beautiful paper that she published in PLOS Biology last year, where she demonstrated C. difficile undergoes this phenotypic switch. And there's these variants in a culture between what's called smooth and rough, as you see here, just because of the morphology. And she did a lot of work to demonstrate how certain genes were important in that those phenotypic variants, as well as some of the implications in the pathogenesis of C. difficile because of this phenotypic switch. But one question that she had in our conversations with her was, what's the role of metabolism play in this setting? And so Matt took his new C. difficile model and integrated some gene expression data that Rita had of the smooth and rough variants. And again, as you can imagine, expression data from some of these colonies or in these complex communities can be a lot easier to generate than metabolomics data specific to each variant. So real quick, and this is my last data slide here, Matt integrated with riptide the expression data associated with these two variants. Okay. First of all, the predictions of the respective growth rates were comparable to what's observed experimentally, and that there's no difference in the growth, which is as observed experimentally. Matt then took those models to predict genes that were essential in one variant versus another. And there's an interesting observation he made is the role that glucose as one substrate and proline as another substrate had. Genes associated with glucose were essential in smooth variant, and genes associated with proline and stickling fermentation-related pathways were essential in the other. So similarly, Matt had predictions with his riptide models for uptake rates of these two different substrates that match. Now, this is a picture that Matt just generated just a few days ago, just to emphasize that this prediction of how critical glucose is as a part of these two different phenotypes. In the absence of glucose, smooth colonies take a more rough shape. You see the more finger-like extensions on the colonies. Matt's going to be repeating this data. Again, this is just brand new in the last few days. But again, to emphasize that this important variant that has important pathogenicity implications of smooth versus rough variants of the C. difficile cultures, that the model helped us point to essential metabolic processes that were critical as a part of this phenotypic switch. So in summary, talked about this ASF community as a model microbiome, some of this work to characterize metabolites that are shared, this new method for gene expression data integration, and how some of these approaches are allowing us to really drive discovery activities and understanding important pathogens associated with microbial communities. I'll just give a little plug here of some training training opportunities at University of Virginia, which is a wonderful place. Grad student applications are coming due in the near future. We have great training in systems biology at University of Virginia, which has a medical center and engineering school and close walking distance between each other. So let me acknowledge those involved. Greg Medlock, again, is a PhD student that won projects, now faculty in pediatrics at UVA. Matt is a postdoc in the lab who drove a lot of the other work that I talked about. The funding that supported the work I mostly focused on here was from the NIH. And then you see some links to our Twitter account and everything else. So Priyanka, Ines, I took like 1 or 2 minutes longer than I was supposed to. I'll stop talking now.
Thank you so much, Jason. This was really enlightening talk. And your group is doing amazing work in the field of microbiome also. This is amazing. And we have a lot of questions from the participants, but we'll take them at the end of the session. I would